Well, welcome everybody to the departmental seminar. It's a pleasure to have everybody here with us today, and it's a particular pleasure to introduce Zach Fuller. Now, I've actually not met Zach before about 2 minutes ago, but he attracted my attention a few months ago as I realized that he's the same guy who was on a paper about coral reef bleaching population genetics in Science last year, and then one on in Nature Genetics on human precision medicine, if you like. And I thought that's a pretty rare combination. Then I just noticed that he won from the Genetic Society of America one of the Editor's Choice Awards for last year for a paper on sex ratio in Drosophila pseudoobscura. So a really eclectic guy, which I think really fits our interests here in the School of Biological Sciences. Certainly, many of us in the genetics community here have started in Drosophila and have moved into different systems. So I'm not going to say much more than that, just to say that welcome, Zach. It's tremendous. We're looking forward to hearing what you have to say, and thanks for coming in the midst of the pandemic, as it were. So over to you.
So I'm Zach Fuller. I'm a postdoc at Columbia. And so I want to start off by perhaps the not too terribly insightful observation that variation in traits surrounds all of us. So some easy examples of this are for obvious traits, such as height in human populations. So for example, if I watch my favorite basketball team, the Denver Nuggets, our center Nikola Jokic is nearly a foot taller than our point guard Jamal Murray. There's also variation in traits that are behavioral. And so I think if one thing that anything has become abundantly clear over the last year or so, it's that humans vary quite a bit in their basic behavior. Lastly, these differences between individuals may be related to traits that underlie disease risk. So for instance, we know that individuals vary in their risk for developing coronary artery disease. So another basic observation is that, like the abundance of variation that we see in traits or phenotypes between individuals, we also see differences between individuals and their genotype. So in a population, if we had the genetic sequence of all individuals and compared each nucleotide at each base pair position in the genome, we'd of course see a number of sites that differ among individuals, and these alleles can occur at different frequencies. These differences are of course originally introduced by mutation, but over generations, these genetic differences are shuffled into new combinations by recombination and are altered in frequency by genetic drift and natural selection. These genetic differences encode the heritable component of differences in traits among individuals, so for the examples that I mentioned, including behavior and disease susceptibility. Because heritable variation reflects the action of genetic and evolutionary mechanisms operating over long timescales in large populations, we can use these patterns of genetic variation to learn about these processes as well as the traits they influence. So these relationships between genetic differences to phenotypic differences have been studied for over a century in model systems that are easily manipulated in the lab, like Drosophila, which is where I spent my Ph.D., or for systems that have a particular relevance to humans, such as humans. So while many broad aspects of these relationships and the evolutionary forces that shape them have been revealed in these systems, there's still much that we don't understand about how heritable variation is shaped by selection and how heritable variation may allow for populations to adapt or underlie deleterious variation or traits that can persist in populations. However, over the last 10 or 20 years, largely fueled by advances in sequencing technology, there have been a couple key breakthroughs in the data that we can now generate that have really transformed what we can learn by studying these patterns of heritable variation. So the first of which is that we now have the ability to generate both genotype and phenotype data for extremely large sample sizes. So already databases like the UK Biobank have detailed phenotype and genotype data for hundreds of thousands of individuals, and available exome data sets are already in the hundreds of thousands and will very soon be in reality where we can now have publicly available sample sizes in the millions. The second breakthrough is that while large-scale genomic sequencing was previously limited to traditional model or clinical systems, we can now cheaply sequence a number of other species more than was possible even a decade ago. And so by studying these patterns of heritable genetic variation and now extremely large sample sizes and across a diverse range of species, some of the fundamental questions that we can now hope to answer using this empirical data are: how do genetics and the environment interact to influence traits? How do populations adapt to changing environments? And what are the typical fitness consequences of mutations? So for the rest of today, I'm going to explore some of these themes, answering questions in two seemingly different systems, in corals and in humans, but in both I'll show how we can use patterns of heritable genetic variation to understand differences in traits, as well as the evolutionary forces that have shaped patterns of this variation. So for the first story, I'll talk about some of my recent work using genetic variation in corals to help understand how populations may adapt to climate change. And so why study corals? Well, coral reefs are vital ecosystems. Despite covering less than 1% of the ocean floor, they support more than 25% of all marine biodiversity. However, recently reef conditions worldwide have been rapidly declining. So I've been interested in using genomics and population genetics to try and better understand and predict these declines. Reefs themselves are primarily made by a group of animals called scleractinian or stony corals. So this is a picture here of a species called Acropora millipora, which is common across the Great Barrier Reef and elsewhere in the Indo-Pacific. Within a colony, individual animals are known as polyps, and they can reproduce asexually through budding to form a multi-polyp colony. These animals can also reproduce sexually, where they have a very unique and interesting life cycle. So they have a mode of reproduction called broadcast spawning, where all individuals annually release gamete bundles, pictured here on the right, which contain both egg and sperm. They're hermaphroditic, although they don't typically self-fertilize. So adults are sedentary, which means, of course, they can't move, with the effect being that adults only reproduce with those in their vicinity. However, once fertilized, the larvae can potentially disperse over much larger geographic distances before they eventually settle. And living inside the host coral tissue are intracellular symbiotic dinoflagellates or algae. And corals only acquire these symbionts once they're juveniles, meaning that they're symbiont-free as larvae. They belong to a family known as Symbiodiniceae, and through photosynthesis, they provide up to 90% of the energy requirements of the coral host. Rust can cause this symbiotic relationship to break down in a process known as bleaching, which leaves the colony a pale white color because all that's visible is now the stony skeleton underneath the translucent polyp tissue. Because the symbiont provides the majority of energy, prolonged periods of bleaching can leave the host coral essentially starved and eventually dead. When entire coral communities experience stress and bleach simultaneously, they undergo something known as mass bleaching. This map shows the average mortality observed on the Great Barrier Reef in 2017. We can see that in some regions, such as here up in the north, more than 2/3 of all corals died as a result of mass bleaching. And the primary culprit of these recent mass bleaching outbreaks is acute heat exposure. This map depicts extreme temperature changes over the Great Barrier Reef in that same time period in 2017. We can see that there's a high correlation between those regions exposed to the most extreme increased temperatures where the most severe losses in coral cover occurred. So although recent mass bleaching episodes have decimated this reef and others worldwide, there's actually quite a bit of phenotypic variation within natural populations. And this can vary both between species and across reefs. So here in this footage, we can see some colonies, and while some appear completely bleached, others, even their immediate neighbors, appear more resilient. So together, I'll argue that these observations and properties of corals, as well as their symbionts, make it a powerful system to study climate change response. So first, we know that bleaching response is an ecologically important trait, and it's established that it's influenced by rising temperatures. So this is a phenotype that we can look at to really understand how climate change impacts species. Number two, we also know that the phenotypic variation in a related trait, thermal tolerance, is at least partially heritable in the coral. It was a motivating reason for me to first look at this genetic variation from the perspective of the coral host. So this is a figure from a 2015 paper from some of my collaborators where they made reciprocal crosses from the adults of the species Acropora millipora collected at 2 different locations on the Great Barrier Reef. And with those individuals collected from Princess Charlotte Bay, shown here in red, experiencing higher temperatures on average and presumably may be adapted to these conditions. And indeed, larvae having a parent from this region survived longer at higher temperatures in stress experiments in the lab. So we can see these larvae in red lines here having a parent from Princess Charlotte Bay, and the time spent at high temperature on the x-axis here. And importantly, this effect was demonstrated in larvae, which again have not yet acquired their symbiont, so we know that this effect is from the coral genetics. Lastly, the insights that we gain using genomics can be used to inform conservation efforts. For example, some of the proposed conservation methods are to selectively breed more tolerant individuals. However, if we currently have no method for identifying these individuals, and if we wait for mass bleaching to reveal them for us, then it's already too late. So the idea is to go take a coral at standard conditions and be able to predict if an individual will bleach or be more tolerant without actually having to expose that individual to increased temperatures. So my motivation for this work was to borrow approaches that have worked well in agriculture as well as humans to see if we can use genomic data to predict which coral individuals are likely to be more tolerant to bleaching. However, these approaches are not possible if we don't have genomic resources in this system. So for example, when I started this work in Acropora millipora, there was no publicly available reference genome. So the first part of the results that I want to share is work towards building this reference genome. So we took a hybrid sequencing approach where we used PacBio long reads as well as Illumina paired-end short reads where we also had 10x genomics barcodes, which gives us some longer range linkage information. So long reads have the benefit of being able to assemble across difficult regions such as repeats, while short reads have the benefit of having much lower error rate. For long read sequencing, we had to extract high molecular weight DNA, which was only available from an adult, and as such, we also ended up sequencing some of the symbiont, which we obviously don't want to include in a coral genome assembly. So to remove symbiont reads and contigs, what we did is we sequenced pools of larvae, which are again aposymbiotic, meaning that they have not yet acquired their symbiont. And so these reads should only be coral, and then we developed an unsupervised classifier to remove these reads. So in other words, if a contig is derived from the coral host in our assembly, we expect reads from the larvae to map to it, while if a contig is derived from the symbiont, we would not be expected to have any larval reads mapping to it. So in the end, we're able to assemble all 14 chromosome sequences of the millipora genome, and it remains the highest quality genome sequence of a coral species assembled to date. We ended up releasing this genome assembly as well as our detailed methods for free through Molly's lab website here at Columbia. And this was about 2 years before we ended up publishing any downstream results on it. And so in that time period, there were over 100 labs internationally that were able to get access to this. All right, so now we have a reference genome, but to start to try and understand how this species may be responding to climate change using genomics, we also need sequences of individuals. So the samples that we ended up sequencing for this work were collected by our collaborator Lena Bay in Australia from about a 300-kilometer stretch of the central Great Barrier Reef in 2017. And for each sample that they collected, we also recorded the bleaching phenotype using 3 different measurements, including a visual score that was recorded at the time of collection. We ended up sequencing over 250 individuals, the majority of which were done at low-fold covered sequencing, where we developed an approach to reliably impute genotypes, which I'm happy to go into more detail later on. At each site, we also had detailed environmental data available for things like salinity, current, speed, and temperature. In total, we had over 40 such environmental measurements that were recorded hourly that we could use to describe the ecological conditions at each reef. And along with the DNA that we extract and sequence from each coral, we also extracted DNA from the symbiont, since again, these are intracellular. And interestingly, multiple species of symbiont can associate with millipora. And by mapping these symbiont reads that we extract to available draft symbiont genomes, we can actually distinguish among different genera of symbionts, such as Cladocopium or C or Durastinium D. And much previous work has shown that these D symbionts are associated with higher thermal tolerance and bleaching resilience. And indeed, we see this effect in our data as well. So this was work that was led by an undergrad who's been working with me and Jihan. And she showed that for samples dominated by these D symbionts, they on average have a higher visual score, indicating that these individuals appear to be less bleached. All right, so now we have a high-quality reference genome and a large sample size of phenotyped and genotyped individuals. So we can finally start to answer some questions now using genomics. So first, we can identify the level of genetic differences across geography and learn about population structure, which may have implications for conservation and management strategies, for instance, by knowing how related or different subpopulations may be. Second, we can identify the functionally important loci involved in traits of interest, such as bleaching, which can help us better understand their biological basis. And lastly, we can use this genomic data to try to predict individual level phenotypes, such as bleaching response. So let's first see what the level of population structure is. So I'm about to show a plot here of a standard measure of genetic differentiation known as FST on the y-axis for each pairwise comparison between a sampled reef site and the geographic distance separating them. So intuitively, this measure scales from 0, meaning that there are no differences between sites, to 1, meaning that there's complete differentiation. So if we expected genetic differentiation to be related with distance here, we'd expect to see a monotonic relationship. So to first orient ourselves, this is what we would see for FST typically in humans for different comparisons and populations. And now here are the data for our corals. So the first observation is that overall we see very low levels of differentiation, where again each point here represents a pairwise comparison between each reef site. And if we zoom in here, we also see that there's no relationship between distance and FST. suggesting that genetic distance is not related to geographic distance across these reef sites, which are spread out over nearly a 300-kilometer area of the central Great Barrier Reef. And this is all presumably a result of its broadcast spawning mode of reproduction, where again, larvae can disperse potentially over large distances. So knowing that there's little population structure has practical implications for conservation efforts. Moreover, this knowledge is also helpful for the next question to address, which is identifying functionally important loci using genome-wide association studies, or GWAS. So one of the lessons learned from GWAS in humans is that the presence of population structure can have a large effect if not controlled for properly, such as causing apparent associations between alleles and phenotype that are actually the result of correlations between environment and ancestry. So this is a simple illustration of the basic idea of a genome-wide association study, or GWAS. So on the bottom, we have what's called a Manhattan plot showing GWAS results, this being a hypothetical example. And so here, each point represents a single variant, and the idea is to go across each variant in the genome and test for an association between genotype and phenotype in the most basic analysis using simple linear regression. And the slope of the line in this test is related to the effect size of a given allele on the trait value. So if under an additive model, this represents the expected increase in bleaching status, for instance, on average for each additional G allele for this particular example. So typically in humans, GWAS requires extremely large sample sizes of thousands or many more individuals to have enough power after correcting for multiple testing to identify loci rising above a significance threshold that we call genome-wide significance. Because for many traits of interest, such as height or schizophrenia risk, they're highly polygenic, meaning that they're influenced by many variants, each with very small effects on their own that are spread throughout the genome. Conversely, in some plant species, such as some early work demonstrated in Arabidopsis, you can actually detect significant associations for particular traits, such as flowering time or bacterial resistance, using similar sample sizes of just a few hundred individuals, because these traits are largely monogenic, influenced by common variants with a large effect. So initially there was a hope or a thought that bleaching response could be similar in corals, influenced by a single large effect locus. However, if we plot our real GWAS results here for bleaching, we see that no single variant rises above a level of genome-wide significance, shown here with this red line. We do see a couple of peaks of variants approaching genome-wide significance, including in some genes that have been previously implicated in bleaching response, such as malate synthase, but it appears that no single variant has a sufficiently large effect size to be significantly associated with bleaching, and our sample size here of a little over 200 individuals. In this GWAS model, we also included covariates to correct for other factors that may influence phenotype, such as, again, the relative proportion of these D symbionts, which we know to be associated with higher thermal tolerance. So at least in the case of Millipora, these results really put to rest the hypothesis that bleaching is influenced by a single gene, and rather that it appears to be polygenic. So while we remain underpowered to detect individual loci associated with bleaching using GWAS, we can take a complementary evolutionary approach to try to identify functionally important loci in the wild by looking for population genetic signatures of where natural selection has acted. And if we think back to this unique life cycle of corals, this gives us a particular clue of what genomic signature to look for. So again, larvae can disperse potentially over large distances while adults can't move. And because environmental conditions vary substantially among reefs, larvae therefore may experience strong selection each generation as they settle on a heterogeneous reefscape. And simulations and models suggest these dynamics of high migration, along with strong spatially varying selection, can lead to a form of selection called balancing selection, where variation in locally beneficial alleles is maintained over unusually long periods of time. And one signal of such balancing selection is high genetic diversity. So we measured pairwise nucleotide diversity in windows across the genome, represented as dots here, to try to find regions of elevated diversity by testing for outliers, which are indicated with red dots. So the more pairwise differences that you have among individuals in a given window, the greater diversity that you have. And remarkably, our strongest signal genome-wide was localized to a single peak on chromosome 7. And if we look at what's underneath this peak, the highest values all fall over a single gene called saxin, which happens to be heat shock co-chaperone. And interestingly, this gene saxin has been shown to be significantly upregulated when exposed to elevated temperatures in the lab in multiple other coral species, indicating that variation in this gene may plausibly be associated with heat response in natural populations. Moreover, in work by a former lab technician and current Ph.D. student who worked with me, Luke, we independently sequenced across this gene region using Sanger amplicons, and we're able to confirm that this signal was not the result from some assembly or bioinformatics error in our de novo genome assembly. So although we see this striking signal of selection at a plausible gene, going back to our GWAS results, those still showed that we don't have sufficient power to detect associated loci given our sample size. But we still wanted to see if we could use our GWAS results to try to predict phenotype. And we can do this using a so-called polygenic score, which is derived from GWAS results. This approach has been very successful in plant breeding genetics, as well as for a number of complex traits in humans. So to construct a polygenic score in the most basic sense, we consider all sites rising above some significance threshold, and then sum the estimated effect sizes for those variants carried by an individual, which then can be used to predict phenotype. So we wanted to see how accurately that we can predict bleaching using both these polygenic scores as well as different combinations of covariates. And so we used a cross-validation approach by predicting trait values for subsets of individuals that we withheld from model training in different partitions. Given that both environmental and genetic factors likely contribute to variation in bleaching, we assessed the prediction accuracy of different linear models using various combinations of predictors. So what I'm going to show here is the accuracy between predicted and actual trait values for bleaching, measured as the squared correlation coefficient, or R squared, here on the y-axis, and then the improvement that we gain by including additional predictors listed on the x-axis. So this first dot represents the R squared for a single cross-validation partition for a model using only the batch effects, which are things like the date the sample was collected and the lane that it was sequenced on. We can then see the improvement that we gain by including genetic principal components, or PCs, which describe the minimal population structure that we observe. And now the environmental variables that we have to describe the ecological conditions of each reef. So far what I've shown is how well that we can predict the bleaching phenotype without incorporating any individual level genetic data. So now I'm going to show the improvement that we get by adding in the relative proportion of D symbionts, which again we infer from our sequencing. So here we see a large increase. And then finally, here's our full model, including all of the previous predictors, along with the PGS, or polygenic score, which is derived just from the host coral genetics. Repeated this cross-validation procedure 100 times, so we can end up comparing the distribution of R squared across these various predictive models. So including the PGS, or polygenic score alone, provides a small yet significant increase in our overall prediction accuracy. In total, this full model explains on average about 62% of the variance in bleaching response. So while we expect our power to increase substantially with larger sample sizes, this signal is certainly promising and in the right direction. We've demonstrated the feasibility, at least as a proof of principle, of being able to predict inter-individual response using both genomic and environmental data. Although in total we only see a small increase in prediction accuracy for the polygenic score alone, we do find that by itself it can actually distinguish the most tolerant individuals from everyone else. So this figure is showing results from the same cross-validation procedure, but using only the polygenic score as the predictor. So the points to pay attention to are these blue dots, which represent the mean trait values for cross-validation partitions. So on the y-axis, we have the actual measured bleaching phenotype, with higher values indicating less bleaching. And on the x-axis, we have deciles of predicted trait values. So the pattern here is that those samples that we predict will be the most tolerant to bleaching do indeed have a significantly higher bleaching score, indicating that they're less bleached. And this is information just using the polygenic score. So this work, I hope, illustrates how we can use genomes and genomics to both understand climate change response and to predict traits related to climate change. So for this specific case of Millipora, we found that there's minimal population structure across our sample locations. We showed that bleaching response is likely polygenic, influenced by many variants, each with small effects on their own. And finally, using the results from GWAS, we demonstrated the feasibility of predicting bleaching response using both genomic and environmental data, including sequencing of both the coral host and of the symbiont. Finally, and perhaps most exciting, is I think this work really opens the door for many more questions. So one of the major limitations of our first study was the relatively small sample size, at least compared to human genomic standards. So the first step that we want to take is to increase the number of individuals that we both sequence and phenotype. So myself, along with our collaborators, were funded by the Agoran Institute to expand our collection past 1,000 individuals, or the hashtag 1K Coral Genome Project. And these collections are already well underway, although I don't think I need to go into too much detail for why this has been delayed over the last year or so. And having this larger sample size will allow us to precisely estimate the heritability of bleaching, which we currently can't do using only a few hundred individuals. It'll increase our power for GWAS and the polygenic score derived from it, as well as provide a true out-of-sample test set, so I won't have to rely on the cross-validation approach that I mentioned a few slides ago. Moreover, we also plan to use improved phenotyping to measure bleaching rather than the visual score that we primarily used for our first study. So here's a picture of tanks on board a research vessel operated by my collaborators in Australia, where they can hold collected coral fragments in these tanks in controlled environments, and then slowly ramp up the temperature, measuring acute bleaching in a much more quantitative approach using these instant heat stress experiments. So that was the end of the first story in corals. And now for the second part of the talk, I want to show some results about inferring the fitness consequences of damaging mutations in humans, but again, here using patterns of heritable genetic variation. And this is work that has largely been in collaboration with a graduate student in the lab who actually just recently passed her defense at IPSITA. So despite remarkable progress in human genetics, there's a large number of genes for which we still don't know their physiological function. And this is relevant for a number of reasons. For example, from a clinical perspective, if a loss of function mutation is identified in a gene, we want to know whether that mutation is harmless or if it may increase the risk of developing disease. Yet if we don't know the function of that gene, this is of course a challenging task. Some recent estimates think that the function of as much as 35% of all genes remain unknown. And for many others, we still don't know much beyond their basic functional category. So one approach to try to learn about the function of a gene is to see what happens when you knock them out or to break their function. So in the fly world, where again I spent my Ph.D., such an approach has been used for decades by geneticists, where they can chemically induce a higher rate of mutations, and then look at the resulting phenotypes in large screens, and then relate this back to the sequence of those individuals and identify if a loss of function mutation occurred in any particular gene. The thing it goes without saying, however, that this forward-style genetics approach at the very least carries some ethical concerns in humans. So instead in humans, one general approach that we could use would be through association tests or GWAS. So for example, if we had a set of patients presenting the same trait of interest or disease, and then a set of controls that don't carry that trait or disease, we could perform a standard case-control GWAS, and then use this as evidence for any associated loci or gene we identify as playing a role in that disease or trait potentially. However, this of course requires extremely large sample sizes of cases and may not work well for very rare variants. Alternatively, we can use patterns of polymorphism of loss-of-function mutations in large populations to look at something called mutational constraint, try to learn about a gene's function. So when I started this work, this was aided by the recent development of large publicly available exome sequence datasets such as EXAC or NOMAD, which have full exome sequences for sample sizes now in the hundreds of thousands of individuals. And the papers that describe these datasets made clear that humans in fact harbor an abundance of loss-of-function mutations, and thus can serve as natural models for the inactivation of different genes. The basic idea of mutational constraint can be illustrated with this analogy to survival bias, using this figure of a plane from World War II, which has now almost become a cliched example in any data science or statistics course. The story goes that during World War II, Allied planes were being shot down frequently, and military leadership obviously wanted to slow this down by reinforcing the armor on them. But because metal was scarce and heavy, you couldn't afford to reinforce the entire plane. So instead, they wanted to examine the pattern of bullet holes on planes that had returned from battle to identify regions or sections which most needed reinforcement. So at first glance, it might seem that you'd want to reinforce those areas with the most bullet holes. However, the statistician Abraham Wald had the key insight that you were only observing damage from planes that had returned from battle and thus had survived. He argued that instead you wanted to reinforce those regions which didn't have any damage, as these were likely the regions where the shot down planes had taken critical damage. So extending this analogy to the human genome, we might think in a gene that's non-essential and likely has no effect on fitness if its function is lost, such as an olfactory receptor, we might observe lots of loss-of-function mutations in large populations. On the other hand, for a gene required for embryonic development, it's unlikely that we'd see any loss-of-function mutations in the entire human population, as these are all individuals that by definition have developed. So invoking this basic principle of survival bias, there have been a number of measures developed over the last several years that have been used to try to identify such regions or genes of mutational constraint. In other words, genes depleted of loss-of-function mutations relative to what we would expect if the gene had no effect on fitness. The one measure in particular, called PLI, has gained widespread popularity, and is one I'll be focusing on for the next few slides. PLI stands for the probability of loss of function intolerance, and it ranges from 0 to 1, with 1 representing genes most constrained or depleted of loss of function variation, and 0 meaning those genes that are least constrained or harboring a greater abundance of loss of function variation. So the authors originally used a cutoff of 0.9 in this score to define genes as being strongly loss of function intolerant. This histogram shows the distribution of PLI for each autosomal gene across the genome. This measure has been used successfully in studies to rank genes based on the severity of fitness consequences of single damaging mutations. It's been successful in identifying the most constrained genes in the genome, those most depleted of loss of function variants, relative to what would be expected if losing that gene had no effect on fitness. PLI has also been shown to have higher values for genes classified as strongly haploinsufficient, that is, genes in which a single loss of function mutation has a discernible phenotypic effect in heterozygotes, compared to those genes which show no evidence of such dosage sensitivity. As a result of this observation, PLI and other measures of constraint have also been increasingly used to try to distinguish between dominant and recessive genes. In other words, trying to identify genes in which loss of function mutations in a single copy leads to disease versus genes in which mutations in both chromosome copies lead to disease, while mutations in only a single copy leave that individual unaffected. So PLI and related measures have increasingly been used to say something about the level of dominance of mutations in a gene. However, if we recast PLI and other scores in a population genetics framework, let's see if we can learn anything about dominance with respect to fitness. So we can understand what measures of mutational constraint like PLI are reflecting if we think of patterns of variation in terms of a population genetics theory called mutation selection balance. So let's say that we have a gene where we can label the wild type allele here as a big A. And a loss of function mutation leads to a disease state of that gene, which we'll label here as D. And so the rate at which loss of function or disease alleles enter the population is governed by this rate U, which we'll call the mutation rate. And so once a loss of function mutation is introduced into the population, over time, the evolutionary forces of natural selection can act to reduce its frequency if it induces a decrease in fitness. In other words, lowers the probability that a carrier of that allele passes it on to the next generation. Additionally, the effects of random sampling in finite populations, which we call genetic drift, can also modulate the frequency of this disease allele over time. So the frequency of this allele that we observe at present in the population, which we'll call Q, reflects a balance between the rate U, in which the mutations are introduced, and then removed by purifying selection and attenuated by genetic drift. So this theory then is given the very creative name of mutation selection balance and was developed by early pioneers in population genetics like Haldane, Wright, and expanded by Ney, among others. So under this theory, and in population genetics, we can write out the relative fitness of individuals carrying the wild type and disease alleles in terms of the parameters S, the selection coefficient, and H, the dominance coefficient. So here S, the selection coefficient, measures the fitness of an individual with 2 disease allele copies relative to the wild type homozygote AA, and H, the dominance coefficient, measures the fitness of the AD heterozygote relative to the selective difference between the 2 homozygotes, such that this parameter HS is the decrease in fitness of this AD heterozygote relative to AA. So we don't need to get too much into the math here, but by writing out the relative fitness of these individuals, early work in population genetics derived that if the dominance coefficient is above 0, so if there are any fitness effects in heterozygotes, the population frequency Q can be approximated by the mutation rate U divided by H times S. So what this means is that the key insight is that if there are any fitness effects in heterozygotes, then deleterious homozygotes become too infrequent in the population for selection to act on them to appreciably affect allele dynamics. In other words, homozygotes for the deleterious disease allele are exceedingly rare, and thus selection acts almost entirely through heterozygotes. So here it's the compound effects of dominance times selections, this joint parameter HS, that primarily influences the frequency of loss of function alleles. So we can perhaps illustrate this point more clearly by using forward population genetic simulations. So what I'm going to show here is the probability surface of observing a specific frequency Q of damaging mutations in some hypothetical gene in terms of the parameters S, the selection coefficient here in the x-axis, and H, the dominance coefficient on the y-axis. So using simulations over a grid of combinations of these two parameters, what I'll show in this example is the probability of, let's say, observing a frequency of 5 loss-of-function mutations in a gene observed in a population of 150,000 individuals. So here's that probability surface, where a darker red indicates a higher probability. And so what we can see is that the probability of observing the specific frequency of loss-of-function mutations is maximized along a fixed ridge of HS values. So more generally, the probability of observing a specific loss-of-function count is maximized along a ridge corresponding to different combinations of H and S that all result in the same HS value. So as a result, you can infer a very strong mutational constraint and be led to conclude haploinsufficiency of a given gene, even when the dominance coefficient H is small, provided that S is sufficiently large. So PLI and other measures of mutational constraint are therefore not indicative of dominance per se. So in the absence of other information, you can't disentangle H and S using patterns of mutational constraint, but only learn about these joint effects HS. So since we can't infer anything about the dominance effects of loss-of-function mutations in a gene using PLI or other measures of constraint, what are the implications? So what I'm going to show here is a simulated gene in a constant population size and the cumulative probability of PLI for this gene, where again PLI ranges from 0, meaning the least constrained, to 1, meaning the most constrained. So this particular cumulative probability is for PLI generated from 1 million simulations using a parameter combination of S equal to 0.1 and H equal to 0.9. Now here the red curve overlaid on this is showing the same cumulative probability distribution in another million simulations, but here flipping these two parameter values for H and S. So while S and H are completely different values, the compound parameter HS is the exact same. And what we see is that the distribution of PLI is essentially identical, again illustrating that we can't learn anything individually about H or S from patterns of mutational constraint, and instead only learn about their joint effects, HS together. So those simulations were using a simple constant population size model. However, we can also model much more realistic scenarios of human demographic history. And when simulating under more plausible demographic models incorporating recent exponential population growth, we see the exact same effects, where the cumulative distributions of PLI are nearly identical for markedly different values of H and S, so long as the joint parameter HS is the same. Thus, these considerations clearly indicate that PLI and patterns of mutational constraint reflect HS jointly. However, PLI and related measures were also not designed to be a direct estimator of HS, which again we can illustrate using simulations. So as I mentioned earlier, since we're in finite populations, the effects of random sampling as the result of genetic drift leads to variance in the observed frequency of loss-of-function mutations. So for a given HS value, the expected observed count of loss-of-function mutations are a distribution rather than a single finite value. In other words, what we observe in data is just one outcome of the evolutionary process, but there's actually a distribution of possible values that could have been generated. So this is the expected distribution of loss-of-function counts in a simulated gene of typical length in the human genome under the parameter combination of S equal to 0.1 and H equal to 0.5, or for a fixed HS value of 0.05. So now let's see what the resulting PLI values would be for these different discrete observed loss-of-function counts for this hypothetical gene. So now overlaid on this plot is what PLI would be in this red line, represented on the y-axis, where again 1 represents the most constrained, and 0 represents the least constrained for these different observed counts of loss-of-function mutations. So again, this is the distribution of loss-of-function counts in a simulated gene under the exact same HS value. And so this illustrates that you can get wildly different PLI values ranging essentially the entire span of this score from 0 to 1, even under the exact same value of HS because of the effects of genetic drift. So this distribution is colored based on if that loss-of-function count yields a PLI value above or below 0.9, which is again what the original authors used to define a gene as being strongly loss-of-function intolerant. So again, in this case, it's quite easy to infer either strongly loss-of-function intolerant or not, even for the exact same value of HS. So thus PLI and other measures of constraint are not direct estimators of HS. So if we really wanted to try to measure HS based on patterns of mutational constraint, what do we want? So we want this estimator to be a direct estimator of HS. Additionally, because of the effects of drift, we want well-calibrated measures of uncertainty. And finally, because the effects of drift change with population size, we also want to incorporate plausible models of human demographic history, particularly for the recent past, as we have evidence that humans have undergone recent explosive population growth. So if we want this estimator that contains these properties, we can use a population genetics framework. And so what are the input data that we can use to try to infer this value? Well, so first we can take the mutation rate U to loss of function mutations in a gene. These per gene mutation rate estimates to loss of function alleles are actually available from a model based on a 2014 paper by Samocha et al. And so these rates U are available for each gene in the genome. Next, we also know that we can get an estimate of Q, the observed frequency of loss of function variants for a gene in a population. So for the observed frequency of loss of function mutations, we can take the frequencies provided by the exome dataset Nomad. And so what I'll talk about here going forward is all data using the largest population present in Nomad, which happens to be non-Finnish Europeans or NFE individuals. And there's about 70,000 of those in Nomad. So in these estimates of Q, we'll use the cumulative frequency of all loss of function variants in a gene. So let's say we have a hypothetical gene and a large number of samples here labeled 1 through n, each with genotype calls that are annotated, so we know what the function of each genotype is, or each base position. And so we'll just consider variants that lead to a loss of function, and simply take the observed frequency as the total cumulative sum of those variants over the number of sequenced chromosomes. So this makes the simplifying assumption that all loss of function mutations in a gene have identical effects on fitness. However, this is plausible, and thus each gene can be modeled as a single biallelic locus with a single mutation rate U and a single frequency Q. So by assuming identical fitness effects for all loss of function mutations in a gene, compound heterozygotes implicitly have the same fitness effect as homozygotes. Finally, we also now have estimates and models of plausible scenarios of human demographic history. We'll call all the parameters that describe this demographic history theta. So for example, this is a model of human demographic history inferred for non-Finnish Europeans, where we have time and generations on the x-axis moving backwards from left to right, and estimates of the effective population size on the y-axis. So in this model of demography, we start in a constant population size, which undergoes growth, followed by a bottleneck, coinciding with the out-of-Africa migration, and then a period of recent strong population growth. So the goal then is to take all of these inputs and then infer a distribution of HS values for a given gene. And so having such a posterior distribution of HS values for a given gene allows us to directly estimate HS, as well as quantify the amount of uncertainty we have with that estimate. So specifically, if we have this distribution that incorporates a demographic model and variance due to drift, we can get well-calibrated measures of uncertainty and define credible intervals of HS values that we believe are likely to have generated the observed frequency of loss of function variants for that particular gene. So to go from this various input data for a gene to a posterior distribution of HS, the approach that I took was standard approximate Bayesian computation, or ABC rejection sampling, as the tool to infer this posterior distribution using these input parameters. So I'm happy to go into much more detail about this method because there's a few more bells and whistles beyond this basic approach. But the general idea is to take a sample of a random HS value from some prior distribution. Here I'm using a log uniform from 10 to the minus 7 to 1. And then take our input parameters and simulate a loss of function frequency using forward population genetic simulations in a Wright-Fisher model over this demographic history using this sampled value of HS. We then keep that HS value if it generates a simulated loss of function frequency that closely matches our observed data Q. So then we repeat this procedure a very large number of times, only keeping those HS values from each iteration that generate simulated frequencies that closely match the observed data. So finally, using the simulated HS values that we keep, we can then generate this posterior distribution. So I use this approach to estimate HS for all autosomal genes in the genome. And so what I'm going to show here is the cumulative probability function for the point estimates of HS, which I'm taking here as the maximum a posteriori estimate, or the mode of this inferred distribution. So this will essentially rank each gene based on its value of HS, shown on the x-axis from left to right, and then show the percentile of that gene in the overall distribution on the y-axis. So first, for about 12% of all genes, the observed frequency of loss of function mutations are completely consistent with an HS of 0 or neutrality, and so we can't infer HS. And so this is represented as the starting point here with this first dot. So now here, each dot represents the mode of the posterior distribution for each autosomal gene in the genome. And this red line represents a cutoff of an HS equal to 10 to the minus 3, which we define as being under strong selection. So first we can see that a large fraction of genes have the most likely estimate of HS as being under considerably strong selection. However, we can also look at the width of those 95% credible intervals to get a sense of the certainty that we have with those estimates. So here are colored horizontal lines representing the width of the 95% credible intervals for each gene, with a darker blue color representing more certainty. So here up in the upper right-hand corner, we have a subset of genes for which we're fairly confident are under strong selection. So if we take an example posterior distribution for one of these genes, it might look like this, where we have narrow credible intervals surrounding our estimate. So we see that we infer around 15% of all genes as being confidently under strong selection, which is actually in line with previous estimates of the proportion of genes that are essential for normal development. However, for other genes, we get quite wide credible intervals. The posterior distribution for one of these genes might look something like this, where although we get a slightly higher density around the mode of this distribution, there is a wide range of HS values that are consistent with the observed frequency of loss-of-function mutations for that particular gene. All right, so we have now what we think are direct estimates of HS, along with reliable measures of uncertainty. But now we want to see if these estimates can actually predict disease risk. And so one question that we might ask is how well do our estimates of HS predict genes associated with developmental disorders? So here we expect patients suffering from severe developmental disease to have loss of function or damaging variants in genes with higher HS values, since those genes should have a large impact on fitness. We can do this by leveraging a recently published data set of de novo mutation data from about 31,000 trios. And this study was specifically focused on de novo mutations, those mutations which are not observed in either mother or father, but arise in the germline and thus are only seen in the child. So what I'll show here is the genes binned by their values of HS that we infer on the x-axis, and on the y-axis, the enrichment or the fold increase in the number of mutations for a given class, what we see relative to what would be expected from chance alone. So this dashed gray line at 1 represents an enrichment of 1, meaning no increase or decrease in the number of de novo variants compared to random chance given the mutation rate. So first I'm showing here the enrichment of synonymous mutations for each bin of HS values. So since synonymous mutations don't alter the amino acid sequence, we don't expect de novo synonymous mutations that change the function of a gene or have a large effect on fitness. And indeed, in this ascertained sample of individuals, we see no enrichment of synonymous mutation for any bin of HS, where again, this dashed gray line represents an enrichment of 1, so no increase or decrease. However, if we plot the same enrichment for loss of function mutations, in this red line here, we get a large enrichment for genes that we infer to be under the strongest selection, consistent with our expectation. We also see a slight enrichment for genes that are under more moderate selection. From these results, we can show that for genes in which we infer to have the greatest effect on fitness, individuals ascertained for developmental disease do see an enrichment of de novo loss-of-function mutations in them. So finally, I want to end with saying that our estimates of HS appear to be sensible and represent direct estimates of the selection coefficient in heterozygotes. Using our inference scheme, we also have well-calibrated measures of uncertainty that capture the variation expected from genetic drift. And so far, our estimates check all the boxes of our original wish list for such an estimator. However, there's one other property that we want for our estimates of HS, and this is to be applicable for all genes. So the plots that I showed previously were just for the genes in the autosomes and did not include any genes from the X chromosome. And this is due to the fact that, of course, X chromosomes are hemizygous in males, and thus can't be modeled using standard Wright-Fisher selection that we used in our original model, which assumes 2 copies of all alleles for all individuals in the total population. So for this reason, previous estimates of constraint, or HS, are also not applicable to X-linked genes. And this is all despite the well-described observation that the X chromosome appears to be enriched for genes involved in developmental disorders. And so we really want to get an estimate for this set of genes. We ended up modifying our simulation framework for X chromosome genes to allow for the effects of hemizygosity, since females who inherit a single copy of a deleterious allele D are heterozygous, and thus the effects of dominance come into play, yet males who inherit a single copy always have that copy exposed to selection. Moreover, since these chromosomes occur in different copy numbers in males and females, we also now need to deal with differences in the germline mutation rate between the 2 sexes, where in the autosomes we can get away with using the sexed average rate, since again these assume that all individuals carry 2 copies of all alleles. So we employed the same inference scheme as before, but now explicitly model the selective differences between the 2 sexes as well as different germline mutation rates. And so here's HS values for all X chromosome genes. And so this is a similar plot as before. But here what we see is we actually infer around 30% of all genes as being confidently under strong selection, which also so happens to be in line with the observation that I mentioned that the X chromosome is enriched for genes associated with severe developmental disease. And so finally, since we have appropriately modeled now direct estimates of HS for the X chromosome as well as for the autosomes, we can also now start to look and test for differences and fitness effects in different compartments in the genome. So what I'll show finally here is the distribution of HS estimates on the y-axis for different categories of genes on the x-axis. Because of the differences in copy number of the X chromosome between males and females, if all genes were expressed at the same level between the two sexes, females would have double the expression level for all genes compared to males. So I don't think it's too hard to imagine that this would lead to some physiological issues. So thus a large subset of genes on the X chromosome undergo something known as X inactivation, where one copy of the gene is randomly silenced. However, other genes can actually escape X inactivation and are expressed from both copies, some of which are thought to underlie sexually dimorphic traits. Other genes on the X are classified as variable, as they may escape inactivation for only a short developmental stage, are randomly silenced in different cells or cell lineages, or show differences in escape status among individuals. And now here are HS estimates for these various categories of X-linked genes, along with one chromosome from the autosomes, chromosome 10 here shown as a comparison. And each dot here represents a single gene, and they're colored darker blue if more than 95% of the total probability mass is above 10 to the minus 3, or what we classify as being under strong selection. So what we see is that for genes that undergo X inactivation and are thus expressed as only a single copy in females, and by default as a single copy in males, they tend to have higher values of HS. So the average HS value for genes that undergo X inactivation is significantly higher than for genes that escape, or for autosomal genes, which both are again expressed in two copies. Additionally, genes that escape X inactivation and are expressed as two copies showed no significant differences in their average HS values than for autosomal genes. So by having now for the first time appropriately modeled estimates for all genes across the genome, both in autosomes and the X chromosome, we can start to look for selective differences between different genomic compartments and different categories of genes. And learning about the fitness effects of different genes will hopefully allow us to better understand their function and potential role in disease. So to summarize, by modeling the frequencies of loss of function variants in a population genetics framework, we now have direct estimates of HS, well-calibrated measures of uncertainty that incorporate recent human demographic history, and importantly, by explicitly modeling the selective differences between X-linked and autosomal genes, these estimates can be made for all genes across the genome. Moreover, we can easily extend this framework to incorporate demographic histories and polymorphism data of other populations, and hopefully this data will soon catch up so we won't only be limited to Europeans. So pulling off this work requires collaborations across many different fields. So I have a number of people to acknowledge and thank for their contributions. So I want to thank all of the people listed here, as well as my sources for funding, and with any remaining time, I'll be happy to take questions.
That was wonderful. Thanks, Zach. Really tremendous. I'm happy to open this up to questions that people can just either put their hand up or just chime in. There's just a few minutes left for questions. Maybe I'll start out. I've got questions across both domains, but let me start with the last section there. So most human geneticists would argue that there's complete additivity, essentially, for all genes, at least at the phenotype level. Can you comment on that and how that influences your modeling of HS and dominance components?
Yep. So we're actually assuming a completely additive model here. So in our inference scheme, we assume the dominance coefficient to be 0.5. So we assume complete additivity. However, we do actually think that there may be subtle fitness effects even in genes that are classified as fully recessive. And so that's something that we're interested in trying to explore further. Because, while we think that most genes are under complete additivity, we also think it's unlikely that some genes are completely recessive as well, and there may be some subtle fitness effects in heterozygotes, and that selection through those individuals may actually be driving population frequencies.
And so that would extend to additivity at the level of fitness, I guess.
Correct. Yep.
That's really, really cool talk, Zach. I really love the pop gen. So one quick question. When you're trying to estimate HS, since the equation deals with mutation rate as well, how did you deal with that? Because some regions have higher mutation rates than others. Did you assume a blanket one? Or I'm guessing with being in Molly's lab, you're like, wait, we can't just use the same value. Uh,
Correct. Yeah, so we use per gene mutation rates,
okay.
which take into account the base composition, so the number of loss of function mutational opportunities in each gene, which is, yeah, influenced by the base composition, but it turns out that that's really just related to the length of a gene.
Okay.
And so, yeah, but we've seen, however, that this mutation rate can describe synonymous variation quite well. And so we think that I think, there's some issues with it, but I think it describes the level of variation well on a per gene basis.
That's great.
So a quick question from the coral portion. So you showed that there's very low FST across locations 10 or 50 or 100 kilometers apart. What about actually within a few meters? So in your sampling, can you sample corals from the same actual dive
Yeah,
and
so
regard
what
them
we
as
saw
independent?
is we actually got, yeah, and so in some cases we actually observed greater diversity within a reef site than between
Good
reef
Lord.
sites. Yeah, so there, and yeah, a fair majority of cases there was, yeah, higher FST within a reef site between individuals than across sites.
Oh, Calvin, please.
Hi, Zach. Very nice talk. I have a question about the second part. So the HS estimation is at the gene level, and you kind of
Yes.
verify your result based on developmental disorder. I'm interested in knowing what about complex trait? Uh, do they tend to enrich for some other complex phenotype?
Yeah, so that's an area of work that we're actively pursuing, like at the moment, is to trying to link our HS estimates with phenotype data from like the UK Biobank, for instance, and trying to infer and like test like the multiplicative fitness model and see if there's fitness effects for the genes that we infer to be under weaker selection or more moderate selection coefficients, and see if that has any relationship to phenotypes in data. But yeah, right now we've just looked at fairly strong, more Mendelian type disorders, just because that's an easy starting point.
Thank you.